My work here in LifeShore started after an initial planning phase in spring, where we conceptualized the study and planned what we were doing. Spring was coming, and we really were very much busy then. And during summer, we were of course mostly busy with maintaining the study, irrigation, and monitoring growth, and all these kind of things. And with the end of the growing season, a lot of our time was taken up by harvesting all the plants that we have been growing. And now, of course, we are pretty busy with preparing samples for analysis. And after that, there will be, of course, a phase where we will be very much busy with interpreting the results, trying to make sense of them and understanding them, and then, of course, reporting and writing. I am a soil scientist, soil chemist, working at the Linnéuniversitetet Department of Biology and Environmental Science. And before that, I have been working at the Swedish University of Agricultural Science in Uppsala. Started working there in 2009. Worked mostly with water quality issues, and also did my PhD there, which was dealing with phosphorus speciation in agricultural soils. An integral part of LifeShore is, of course, not only this environmental friendly take-up of these sediments, it is also an aim to refer those sediments to a beneficial use, or to recycle them, basically. And one of several options to do that is to use these sediments to grow plants. And this is what my work has been focusing on, and also because I think that this is a very promising approach to recycle these nutrients. So using these sediments to grow maybe edible plants offers an additional, basically, option to contribute to closing this nutrient cycle within food production, and by that making food production more sustainable, and also combating eutrophication, for instance, in the Baltic Sea. This has been the stem of a sunflower growing in one of these substrate mixtures that we included in the experiment, and we do that separating different plant parts and sampling and analyzing them separately, because we want to get an idea of which part of the plants tend to accumulate toxic metals. The next step after preparing the samples is, of course, to set them away for analysis, and that means both plant samples and sediment and substrate samples. And we are, for instance, interested with regard to the sediment in which chemical forms, nutrients and contaminants are present in these sediments. And with regard to these plant samples, we are interested in how much and which metals have been taken up by which plant, and also to get an indication whether those treatments that we have, for instance the addition of biocoal, helped to reduce plant uptake of toxic metals.